So it's really exciting to be with this great audience of energetic and talented women and discuss our work here. As we're setting up, I just want to do a quick introduction. I graduated a double major in computer science and biology from Smith College. It's a women's college. Then I did my PhD from Stanford in the Biomedical Informatics program. I also got a PhD minor at Stanford. I've been at Dana-Farber since 2002. There's some bad news within the audience. In fact, with the whole population, a third of people will be hit with cancer in your lifetime. And traditionally, people, oh sorry, this is my disclosure slide. And so traditionally, when people are curing cancer, they use not very effective approaches. They cut, they poison, they burn, right? Recently, precision medicine is really big. In fact, Dana-Farber, with every patient coming to the hospital, if they are diagnosed, they will get cells and spare the normal cells. Unfortunately, many mutations do not have a known drug that can be targeting the mutation. In addition, a lot of these targeted drugs only work for a few months to a few years, and eventually the cancer will become resistant to the drug. And the really exciting news is in 2013, Science rated cancer immunotherapy as a breakthrough of the year. And in 2018, the developers of cancer immunotherapy won the Nobel Prize. And so I want to very quickly show you the mechanism for cancer immunotherapy, at least the current checkpoint inhibitor antibodies, how do they work? So within our body, all of our normal cells, well, so T cells and B cells are actually like guards in our body. And they have receptors on the surface of the cell. And all the normal cells in our body routinely bring all the degraded proteins onto the cell surface for the immune cells to examine. And so if the presented cell is all normal, then it's okay, it's being allowed to pass. However, if there are mutations in the cancer cells, they will be presented onto the cell surface, and our immune cell will know this is not my cell, there's something going on that's wrong. And normally, the immune cell will be able to kill the cell that has the mutations. Unfortunately, as people age or their immune system become weaker, sometimes the cancer cell is allowed to coexist with the immune cells, especially when the immune cells is learning to deal with this situation by secreting proteins or ligands which can interact with other receptors on the T cells to make the T cell dysfunctional. You can imagine this as bringing up some alcohol to make the guard drunk, right? And so very often in the tumors, you do see the immune cells occur, like they do locate in the cancer cells, but they don't do the killing. And so the current immunotherapy antibodies work in that they can block the receptors, basically kind of wake up the immune receptor, the guards, and then the B cells and T cells will be back to working again, and they very quickly recognize the cancers that have a lot of different mutations as foreign, and then can eliminate the cancer cells. So it's a very different mechanism. It doesn't directly target cancer. Instead, it's trying to boost the immune system to kill the cancer cells. And so this immunotherapy has really shown remarkable efficacy. And really, in metastatic tumors, the lucky patient can have prolonged response and also completely cured from cancer. Unfortunately, only a minority of cancer types and only a minority of patients would respond to the immunotherapy. And so there's vast research on how to predict which patients would respond. And there are some known biomarkers, right? So one is, how many T cells do you see in the tumor? That's kind of measuring how many guards are present in the tumor. The second is to measure the T cell diversity. Do you have different type of guards that recognize different type of foreign objects or mutations? And the third is the mutation load. How foreign is this tumor? In fact, the tumors that have the highest mutation load with many mutations, traditionally those are harder to cure because they can evolve resistance faster, but they are actually the easiest to respond to immunotherapy because they look very foreign. And also the PD-L1 level, which is this ligand that cancer cells bring up in order to get the T cell drunk. Right? So the PD-L1 levels is, you're trying to see, do you see bottles on the floor? There's some attempt for the immune system to at least try to do the attack before the cancer cell responds. And the question is are there additional response biomarkers involved. So that's when our group got interested in cancer immunology. And we first look at the tumor profiles that are publicly available. Very often those tumors are profiled in the bulk. You get a whole piece of tumor tissue. But in there are cancer cells, there are some normal cells, there are all sorts of immune cells. The question is, based on this bulk data, can we do computational deconvolution to figure out how many different immune cell types are there? And we have developed a computational algorithm called Timer for that. In addition, we have analyzed over 11,000 tumor samples to see when the patient have higher expression of certain genes, would that make the T cell infiltrate the tumor better or worse, or other immune cells infiltrating the tumor better or worse, and whether that would really influence patient survival in the clinical setting. These are not necessarily patients that are treated with immunotherapy. In fact, whether it's chemotherapy, radiation, or targeted therapy, our whole immune system always play a role in how those patients would respond to these other therapies, which can give us insights on how our immune system works. Another algorithm we have worked on is really directly predicting before a patient is given the immunotherapy, how likely they are going to benefit from this therapy. And so again, we learn from the patients that are not treated with immunotherapy to see what are the factors that might be important. And so far, there are two scenarios where the patient may not really respond well. The first type is basically this is immune system that's able to, or T cells that are able to infiltrate the tumor. And those are called the immune hot tumor. T cells are there, however, maybe they're too drunk. They are like dead drunk in there, so you can't really wake it up very well. So we have a gene expression signature on the tumor to really measure how dysfunctional the T cells are in the tumor. And those patients, so basically the T cells are just tipsy. Maybe you can use immunotherapy to wake it up, but when it's really dead drunk, then it's a lot harder. The other situation is there are other cells in the tumor that's suppressing the T cell from infiltrating the tumor. Those are actually much, much harder because basically you have a guard repellent. They are secreting different proteins or cytokines, which prevents the T cell from going into the tumor. And those patients usually don't respond to current immunotherapy and potentially need to be treated with something else to get the T cell in before you can wake it up. And so we developed this TIDE algorithm to really use gene expression in the tumors to really measure the signatures for the T cell dysfunction and also the T cell exclusion signatures. And since then, we have been awarded a NCI Cancer Moonshot grant. Basically, NCI is looking at all the immunotherapy trials and doing tumor profiling on those. And we're also collecting a lot of the published tumor immunotherapy data with the goal of really using public data to teach people how to evaluate or develop a better biomarker. And also, if they have patients, how can you use many different biomarkers to predict whether this patient should be treated with immunotherapy or maybe they should be treated with something else. Of course, in order to really do immunotherapy well, it's possible that one drug, the lucky few that will benefit just from a single drug, that's really great, but it's only a minority of patients. In order for immunotherapy to really have wide application, we probably need a really novel and personalized combination therapy. And so for that, we turn to the bacterial immune system. And so you were wondering, like, bacteria, does it have immune system? It's a single cell, very simple, right? In fact, the discovery on CRISPR-Cas9 is a really revolutionary discovery, and the CRISPR-Cas9 is the bacteria adaptive immune system. Basically, the bacteria, there are these little proteins you can think about as the little guards in the bacteria cell, and they take signature RNA, which is a photo of a foreign object, say a viral or some other foreign DNA that they know is not part of itself. And then it will go to a specific location. Well, it will actually circle around the cell to see whether there are DNA that look like this photo, and it will just cut it. And so when this system is introduced into the human cell, basically scientists realize that all you need to introduce is this guard protein, which is the Cas9 protein, and also a piece of photo, which is this guide RNA. It will go into the cell and look for DNA sequence that really match to the DNA sequence and cut when there is a match. And so you can go almost anywhere in the cell and make a cut. And if this happen to cut on the gene of a human cell, very often it will create a gene knockout, so the gene will lose its function. And so this is a really, really powerful technology. You sequence the photos, and then it's like this. For example, initially, say we want to knock out one gene and another gene. To make sure that we really knock out the right guy and not have off-target effect, we show different photos, right? So initially, this is if we want to knock out two genes, we have even representation. But if you knock out the gene, let the cell grow a little bit, and you sequence again, you count the photos, you might notice, the Washington gene is very important. If you get rid of the gene, the cells are dying. And so 2 weeks later, when you sequence, there are not so many cells still carrying this Washington gene left. So the photo is actually a gene that's being knocked out. And so if you don't see this photo very often, it means that this gene is very, very important. When you knock out that gene, the cell is dying or not growing as fast. Whereas if, say, there's another gene that normally is suppressing the cell from growing, and now you knock it out, the cell is growing much, much faster. And so at the end, when you sequence it, you will see a lot of representation of that photo. And so basically, this is just two genes showing you how the CRISPR screening experiments are done. We actually do this experiment genome-wide by targeting 20,000 genes in one experiment, and then just use high-throughput sequencing to really figure out the hits. And of course, for that, you need computational algorithm as well. And we're also using this in an immune setting. Basically, you can have just cancer cells growing, and you treat the cancer cell with all sorts of immune signals, toxic chemicals or proteins that the immune system can release when it detects something is foreign. It's like throwing the cancer cells some bombs and see how they respond. The second one is co-culture. We are co-culturing the cancer cell with the immune cell. And so this is like, you look at all the, the jet plane, not only they can throw bombs, they can also shoot guns, right? So it's a little bit more potent. And the third situation is we do this experiment in mouse. So of course in the mouse system, the immune system is much more complicated, right? There's the Navy, there's the Air Force, and so basically each cancer cell have a different gene knockout. We put this cancer cell in the mouse and then treat the mice with immunotherapy. And so in general, we're giving the cancer cell different type of immune pressure to see how they would respond. And with this, it's actually giving us a lot of insights on how the cancer cell, what kind of genes are good indications of the cancer cell that's going to respond well to immunotherapy. And so not only we have good biomarkers, we also can see if you get rid of some gene, would the cancer cell respond better to immunotherapy? So we can also get good regulators of those immunotherapy response. And you can see for all of this, we are generating a lot of high-throughput sequencing data, comparing different conditions in order to really find out what are the hits. In fact, we need computational algorithm even to know whether the experiment is working or not, right? Whether there are hits, does it make sense in light of all the other public data to really identify a good biomarker and good regulators to improve the current immunotherapy. And once we have a particular hit, we do a genome-wide experiment, we see one hit. Very often with that one hit, what we do is we knock out that gene and then grow the tumor in the mouse and treat the mice with some immunotherapy, and then ask, with or without the gene, do you see the tumor microenvironment changing in terms of this is done using the single-cell tumor profiling. You can look at whether the immune cell populations are changing, and also whether the genes are changed in the same immune cell types after the gene knockout. Right? Does that gene really influence the immune attack to the tumor? And with this, we also use a lot of public data on gene regulations based on the differential gene expressions to figure out what genes are really important to show that gene expression change, to regulate that gene expression change, to figure out the mechanism of the target hit that we identified from the CRISPR screens. And with those, we usually can get a very good target that potentially, if it has a drug already, then it suggests that people should try this new gene with the current immunotherapy to improve immunotherapy response. But also, if this is a new target, we could potentially develop a new drug. And for that, we again turn to the tumor. So we mentioned early on, our immune system is able to recognize all sorts of foreign things, and you wonder, we only have 2 copies of DNA genome. How can we recognize different things? It's because in our immune system, T cells and B cells go through a VDJ recombination. It's able to create some random sequences on the T cells and B cell receptors that can recognize all sorts of non-self particles, or proteins, or lipids, or DNA. And so there are experiments that really profile your T cells and B cells for those type of receptors, but they're actually very expensive. What we are looking at is whether in the patient tumors we can already see the T cells and B cell sequences in there. Unfortunately, with this very hypervariable sequence, it's not in the reference genome. And so all the sequences that really map to the T cell and B cell receptor sequence, when you get the high-throughput sequencing for the genome, the first step is you map it to the genome. And all of these really interesting sequences are thrown into the trash pile as unrecognizable. They can't find a place in the genome because of this mutation. And so what we have developed an algorithm for is to really go back to the trash pile and really ask, can we de novo assemble the T cell receptor and B cell receptor sequences out? And from the public tumor sequencing data, we are able to actually assemble a lot of the T cell and B cell receptors. These are the important guards that are in the tumor and recognizing the mutations and doing the killing job, right? So that's really important. In fact, from this analysis, we found that patient tumors actually contain antibodies that have therapeutic effect. The question is, which antibody is recognizing which antigens, right, in which cancer types. So we're developing machine learning models to really identify the tumor-specific or antigen-specific receptors in order to develop cancer vaccines or CAR-Ts or also therapeutic antibodies. So I hope you can see with computational tools, not only we can predict which patient respond, we can identify biomarkers, we can identify novel regulators, and when we have a good target, we can also use computational method to develop drugs. All of these are possible now with data science. And so in the future, for personalized therapy, I think targeted therapy are still very, very important. They work by directly targeting the cancer cell. Immunotherapy, as we mentioned, is you try to boost the patient's immune system in order to indirectly kill the cancer cells. I'm the director for the Functional Cancer Epigenetics Center at Dana-Farber. We also work on epigenetics. This is the type of gene that really kind of maintain the system balance, and they are very frequently mutated in cancer, and there are a lot of small molecule inhibitors developed against epigenetic mutations as well. Those kind of drugs don't work as well as single drug, but we suspect that as combination, it would work much, much better. But in order to use these different drugs well for different patients, for different cancer types, we really need to have a lot more tumor profile, whether at the bulk level or at the single cell level. We also need to do more CRISPR screens because they really give us a functional knockout or functional readout. If you get rid of this gene, what is the consequence? It's not just an association. And of course, all of this generates a massive amount of data, and we will need computational, statistical methods and tools in order to really make sense of this. And we hope to do this well. The NCI Cancer Moonshot set a very, very ambitious goal for curing cancer this year. I think we'll need really talented women in this audience and really worldwide to work hard and hopefully within our lifetime really cure most of the cancers. So I want to thank members in my lab. Many of them have already graduated and started their own labs. And also, I want to thank our collaborators within the Dana-Farber and at Harvard and also in other institutions. This is really a collaborative effort, and also I want to thank the funding agencies. And these are some of the computational resources that are developed in our lab, which might be useful if you're interested. Thank you very much. Very good question. Currently, there are massive tumor profiling efforts in every hospital, research institutions. The key is the data reuse and the data sharing. And so for that, I think a lot of efforts are being made, but I think central resources are definitely important. This is especially problematic for immunotherapy. a lot of trials are done, the data is available, but data sharing is not so great. And so we are actually working with the American Association of Cancer Research and NCI to create these data central repositories so that people can use public data to help them predict future patient responses.
Thanks, that was a great talk. Kind of along those lines, I was curious, when you were talking about like doing the sequencing, it looked like if you were to do that for the case where it was in vitro versus the in vivo versus in the mouse model, I would imagine that that would be a lot more time intensive to do the mouse model maybe, and I was wondering if you had some sort of strategic workflow associated with strategizing when you would use experiments of different.
Yeah, that's a really good question. So the cost for doing an experiment in Petri dish is lower and it's faster. And for that, we usually screen all the genes in the genome. And by the time we go to the mouse, it's usually only a few hundred to maybe only 1,000 genes. And then, of course, with that result, we also look public clinical data, and there it's much, much harder. In fact, each clinical trial on each patient case costs about $200,000. So there you really have to narrow down your hits. And that's why we use computational approaches initially from a lot of data to gradually help us prune and prioritize. We do have computational pipelines to select which genes do we put in the mouse. And later on, of course, when you develop a drug, it's one at a time, right? You can't really do genome-wide.
Thank you for the great talk. Actually, last year, in our lab, we were looking at into the pathway of the T cell and the same learning, and trying to use the epigenetics or some of the models to simulate that. But actually, when we're doing this, we have a very hard. It's very hard to get a very solid data, or we can't like. We don't have the criteria to evaluate what kind of the data is qualified for our simulation. So I wonder if you have.
So the question is, how do you decide which data to use, right? So,
or we're not sure if because they have a lot of like assumptions from the lab, from the biology lab.
So I would say metadata for annotating the data is very important for reuse and also quality control. Even high-profile published papers have garbage data, definitely. So we have to have good computational methods to really to decide do we want to include this. And also what we do find that in clinical cohort, every cohort is different. It could be the hospital, could be their profiling approach, and there are definitely some cohort-specific differences. That's why the more cohort you have, the more confident you are. You will never see any biomarker that works in every single trial. And so it's like, oh, if this thing works in maybe 80% of the trial, that's already phenomenal. And fortunately, so far, most of the biomarkers were developed on 2 trials with very handful of patients. And so that's why we really hope that people who are doing those trials will make the data available for the community to learn.
Yeah, the ability is very important, but also because the computational model is kind of like talking about the general in the universal idea, but it seems like the data is always like case by case.
Mm-hmm. Yeah, because, well, so I would say right now, tumor profiles are much, much cheaper compared to a clinical trial. Clinical trials, I mentioned, is $200,000 a piece, right? Whereas tumor profiling for things like RNA sequencing, you can do this with less than $200 a sample. I would think that people should always do RNA sequencing now. It's just so informative, right? And if the data can be pulled with the metadata annotation available, it will do a lot of good for the community. Is there another question in the back?
Thanks for such a great talk. One question around sort of sample sizes and that sort of thing. As far as getting enough data, and sample sizes versus kind of personalized medicine that's supposed to have an N of 1, I can see mathematically how these are at odds with each other. Have you been able to sort of make sense of that from a biological standpoint?
Yeah, very good question. If you don't have enough data, how can you make a confident prediction on when? This is a key aspect of what we are trying to learn. As I mentioned that whether a patient is treated with immunotherapy, even if they are treated with chemo, radiation, or target therapy, immune always play a role. And so that's why we are trying, right now the immunotherapy trial samples are still very limited. We're trying to use the non-immunotherapy data, all the mouse model data, all the cancer cell line data, anything that you can learn from the public to help us prune so that you can narrow down on a few hypotheses. Then maybe you can look at the result in individual patients. And also, yeah, as I mentioned again, tumor profiling, you think, actually TCGA project from the NCI has finished. People think, okay, we have about a few hundred cancer types, a few hundred samples for each tumor. That should be sufficient to characterize the tumor. But when you really want to know, I want to look at breast cancer, HER2 positive, that are resistant to this drug, then there's like 3 patients left. So it's still not enough.
Thank you very much for this great talk. I have one question come up to my mind with this data quality. And how do you guarantee or maybe kind of estimate the reliability of your result? Are you taking all the data available or you have some kind of filters?
We do try to use all the available data, but we do a lot of work QCing, checking the metadata, really deciding whether to use a particular public data resource. And then for immunotherapy, as I mentioned, the number of cohorts that do have available results are increasing right now. And so you can train your models, and then hopefully on the new cohort, so far it's interesting, within cohort course validation, usually it's easy. The difficulty is when you train a biomarker and you test it on a new trial, that's actually a really good test. And that's why we're hoping more and more new trials will be available. We can reevaluate all the existing biomarkers.
Any additional questions before we move to our next speaker? Thank you so much, Professor.